disease, some tests problems. So all these things are the diseases for which how can we go in for the solution. Today we are going in for allopathic treatment and too many types of treatments, varieties we are going in to eradicate such diseases. Can we eradicate such diseases nowadays? There are diseases which are so-called the eradicated diseases. They are number 1 leprosy, then polio, then any other diseases which are eradicated, smallpox. So these are all the so-called eradicated diseases. But till now there are representative cases are there in and around areas, in districts, in our state or somewhere in the world. Okay. So we cannot eradicate any one of the diseases. That's why a school of scientists, they sit together and think about the genomes of humans, so that once we touch the genes, once we correct the proper DNA which is responsible for a disease, that will be touched and that will be corrected, so that we won't get that disease in the future generation. Since it's a very good idea, all the school of scientists, they thought that they can consider this this is a human genome project, and they are in need of some money for going ahead with this project. They are in need of some scientists from all over the world. Since it's a multinational project, scientists and funding agencies must be from different nations of the world. So they have to collect the scientists and laboratories. Accordingly, this is a human genome project is coming under Human Genome Organization. That is, in short, it is called HUGO. Then there are funding agencies. People are also much interested, the public are much interested, and they are contributing their own contributions to this project. Apart from the funding agencies, just like the Department of Energy, European Commission, what else? Sanger Institute of Japan, the National Institute of Health from United States of America. So all these countries are grouped together for contributing to this project for its successful outcome. Okay. As a result, what happened? So up to this, with this I can conclude this introduction of this project. How can we go ahead with this work? That is methodology. How can we go ahead with the methodology, the methods to execute this project? The first is we have to separate the DNA, because DNA is involved here. Genome is involved here. Where is genome? What is genome then? What is genome? It is a group of genes, that's all. Where is genes then? It is in the DNA. Where is DNA then? It is in the chromosome. Where is chromosome? In the nucleolus, then in the nucleus, then in a cell. Okay. This is the whereabouts of the genes. Okay. So we have to separate this DNA from the somatic cells. We are not in need of the gametic cells, which is in somatic cells. We have to separate it. Now let's follow the flow chart one by one, how to get this DNA, separate the DNA from the somatic cells. First, what we have to do is, we have to make a cell line culture. This cell line culture is homogenated with the help of this sucrose solution. By adding sucrose solution, then it is subjected to differential centrifugation. After differential centrifugation, what happens? What we will get usually when we are centrifuging something, we will get a supernatant and a sediment. Some sediment underneath. So we are discarding the supernatant and taking only the sediment. From the sediment, we are adding once again the lysis buffer. Why? For it is subjected to ultracentrifugation. After ultra is very fine. After ultracentrifugation, what happens? We have to get a sediment. We have to decant the supernatant and take the sediment. From the sediment, we have to store it in minus 20 degrees centigrade in the refrigerator for future use of sequencing the genes. Okay. How can we sequencing the genes? That is our next step. How can we go ahead with this sequencing? Because this DNA, what we have got, it must be cut into pieces first. How can we cut into pieces? Using scissors? Restriction